here can get our review into. Okay, here's the review. Let's see. Without your notes, who can tell me the name of the teeny tiny artery that takes blood into a glomerulus? Give me a little thumbs up or raise a hand. Peyton's got a guess. What do you think, Peyton?
Is it the efferent artery?
The afferent arterial. You got it. I saw your thumbs up too, Jacob Krogman. Sorry about that. Yeah, afferent arterial. Very good. Let's see. So once we're in this little capillary called a glomerulus, there is a capsule that surrounds it. Who thinks they know the name of that capsule off the top of their head? Mr. McIntyre, what do you think?
Bowman's capsule.
Bowman's capsule. Good answer. Good answer. All right. Let's see. Who thinks they know the term for the outer layer of a kidney? Meaning there's an inside. If you cut it in half, there's an inside and an outside. Who thinks they know the outside? Mr. Krogman got thumbs up.
Cortex?
The cortex. And what would the inside be?
Is it the medulla?
The medulla. Very good. John Dobbs would like to add you as a contact. I don't know what that means. Zoom? Sure.
I don't.
Go for it, John. I don't know what that.
Now
Yeah.
I'm confused. What is
That's
going
cool.
on here?
You can contact me. Okay. Yeah. All right. So kidney, kind of shaped like a kidney bean, or is a kidney bean shaped like a kidney? It's really hard to make a kidney with your hands. Never tried to do that. There. Oh, that looks pretty not kidney-like. Okay. There's a concave side where all like the blood and arteries and veins and ureter enter. What's that little concave side of the kidney called? Mr. Peyton's got a thumbs up. What do you think, Peyton?
Is it like the hilus?
It is the hilus. Yeah, the hilus or the hilum sometimes it's called. That's a real good answer. Let's see. What else should we do? I'll give you a real hard one. What is the functional unit of the kidney? There's about 1 million of them in each kidney, and it's kind of a series of little tubes. Mr. McIntyre wants to guess. What do you think, Jake?
The nephron.
Good guess. Good guess. The nephron. Okay, you guys you got this kidney thing down. That's real good. Okay, cool. So I'm going to share my screen, and we'll get back to it here. Oh, let's see. Share desktop. Yep, that'd be a good place to start. Okay. So you're all seeing my PowerPoint now, I hope. Little nephron deal in there with blood going in and out of it. We talked about peritubular capillaries last time. We talked about the afferent and efferent arterial. Today we're going to get to talk a little bit about how the blood flow works between here and here and how we get blood plasma out, which is pretty incredible. And I don't really claim to understand every little piece of it because it's pretty amazing, but that's going to be kind of a fun deal. So let's talk about nephron. Nephron, Jake was absolutely right with that. The nephron is the functional unit of the kidney. It's where we get words like epinephrine. Nephron essentially means the way the kidney is doing its job. Each kidney's got about 1 million nephrons. You probably know at this point that you can get by with one kidney. People donate kidneys all the time for people who are in kidney failure, and you can filter all the blood you need to filter really with one kidney. Nephrons are microscopic. They're very, very small, and they exist both in the medulla and the cortex. Now, that doesn't mean that it's like there's nephrons in the cortex and there's nephrons in the medulla. No, it's like where the renal pyramid touches the cortex. The nephron starts in the cortex, loop of Henle into the medulla, loop of Henle back out into the cortex is kind of more what that means. So they're right on that interface between the inside and the outside. They're made of two parts. I don't really care that you memorize renal corpuscle and renal tubule. The corpuscle is basically the Bowman's capsule. I'd rather have you know Bowman's capsule. And then the tubule is the loop of Henle and the collecting duct. And so these guys, you don't have to put those in your brain forever, but that's the nephron. We're going to look at a more detailed picture. I want you to know this picture of the nephron. Corpuscle, tubule, that's a little overly simplistic. I'd rather have you know Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct. That would be where I would want you to be there. So that's a nephron. Everybody got questions on that, or we need a little minute to write that down? No? Cool. All right. Teachers only wait a second and a half on average before they respond to something when they ask a question. So I want to make sure that I embrace my teacher stereotype and say, any questions? Nope. Good. Great. Okay. Moving on. Bowman's capsule. You should all know Bowman's capsule at this point. Bowman's capsule is what surrounds that glomerulus, that capillary network is the glomerulus, and Bowman's capsule goes around that. And we're going to push that fluid, the blood plasma, out of the capillaries, and it's going to collect in the Bowman's capsule. And this is essentially where filtration and reabsorption and all of that kind of stuff is going to start. Glomerulus, fun to say, fun to spell, is the capillary network, and that comes from Latin for tiny ball of yarn, which kind of makes sense when you think about what the glomerulus looks like. It's a little bunch of tiny threads, which are the capillary bed. And you guys are familiar with capillary beds from our cardiovascular unit. So here's the new thing. The capillary bed is surrounded by little cells called podocytes. Okay, podocytes are little cells that basically have filtration slits in between them. And so I'll show you a picture of this in a second, but essentially your capillary bed is, here there's one itty bitty teeny tiny vein, right? That's a capillary. And then there's this cell on top of it, right, called a podocyte. And the podocyte has a filtration slit with the podocyte next to it. And there's so much pressure in here that fluid can leak out. But the filtration slits are so small that red blood cells cruising along here cannot leak out. And so podocytes are the way we're going to get filtration accomplished. Well, that's not really true. Podocytes are the way we're going to get, how should we say this? Separation of blood plasma and formed elements in your blood. We're going to get the fluid piece out of the bloodstream, out of the capillary. We're going to keep red blood cells, white blood cells, platelets in your actual arteriole there, your capillary bed, which is kind of cool. And so that's podocytes are very, very important for that. That's their job. All right, a much better picture. Here we have, did I click too quickly? Let me know if I need to go back. Nope, you're good? Okay, cool. I try to babble a bit to give you time to write, So anyways, afferent arteriole is this guy. Here's the blood coming in. This is a really cool picture because it comes in, goes through this capillary network. So the blood is flowing through here, right? Real slow. These are podocytes just completely covering the capillary. They're in purple. And all of these little lines right here that you see, those are filtration slits that are going to cause that blood plasma to be pushed out, to leak out. And as that leaks out of these holes, it gets captured in this Bowman's capsule and literally just flows from the Bowman's capsule to the proximal convoluted tubule. And there's lots of things that can be pushed out of this area. Primarily, we're talking about fluids and dissolved salts, but it kind of depends. You could get glucose could be pushed out if you're diabetic. If you eat a ton of vegetation, you can actually have vegetable fibers, small vegetable fibers get pushed out of these podocytes. And the only reason I know that, actually, this is weird because it's going on YouTube, but whatever, it's too late now. I mean, I tell you guys weird stories in anatomy, so I'm going to keep telling you weird stories. Long time ago when I was in college, right, just a young whippersnapper like you, we had to do a physiology lab on the urinary system. And I was thinking, that sounds terrible. How are we going to do a lab on the urinary system? And they said the only thing you need to do for the urinary system is save your pee. Don't pee in the morning. Get up and come to the lab. And I was just thinking, this can't be good. There's no way this is going to be good, right? And so there were a bunch of labs. This is a 3-hour lab, right? This is a long time to work with urine. And one of the experiments was, and we just had to do terrible things. Like you had to run laps and sweat and then collect your pee and bring it back to your group and then run more laps and collect your pee and bring it back to the group. And I'm thinking, this is not what I signed up for. But one of the experiments was somebody had to go to the bathroom, bring back a sample of urine, and you had to put it under a microscope. And I kid you not, one of the people, they had this list of things you were identifying. You could test it for glucose, and you had to look under a microscope. And one of them had long green fibers in their urine. And we're like, what is this? Is this wrong? What is this? And I guess the person had eaten, like for a snack, they'd eaten like an entire bag of sugar snap peas. Like that was their snack. They just ate peas and literally microscopic plant fibers were coming out in their urine. I was like, I can't believe those got through. Like, how did they get through? That's crazy. So yeah, I mean, lots of stuff can get through filtration slits apparently, because who would have thought? So that's why we don't do labs in the urinary system. Dissecting kidneys are awesome. Urinary labs are disgusting. But hey, you know, whatever. It makes for a good story, I guess. So off the blood plasma goes into the proximal tubule, where we're going to immediately start reabsorbing tons of water with our peritubular capillaries. So you'll also notice, this is kind of a cool thing. Let's get really fancy and make this pen blue. That'd be kind of neat. Pen color, blue. Okay, so as the blood has flown through these capillaries and is on its way back out, you'll notice that our efferent arteriole is actually a little smaller in diameter than our afferent arteriole was. Any physics nerds or plumbers want to make a guess as to why that might be? I shouldn't say physics nerds. This is going on YouTube. Dang it. Too late. Can't back off. Come on, physics nerds. It's a compliment. Being nerdy is a compliment. Guess. Make a guess. Why would the outflow be a smaller tube than the inflow?
Less pressure.
Mm-hmm. So you think about this like you put your thumb over the end of a garden hose, right? And it like sprays out further. So this is like six lanes of traffic coming in this way with the blood this way, right? And the outflow, right, the efferent arteriole is like they hit a construction zone. Okay, so we just took 6 lanes of traffic to 2 lanes of traffic. So there's going to be a huge back pressure in this capillary bed. And that back pressure is what's going to cause blood plasma to actually get pushed out of the bloodstream. Because if you think about it, if blood flowed through the afferent arteriole, down through the capillaries and out through the efferent arteriole, and it was the same pressure the whole way through, there's no way we could push plasma out of the blood. It'd be at the same pressure that your blood pressure is at normally in all your other arteries and veins. And we're certainly not leaking blood plasma into all of our organs and muscles. That's ridiculous, right? But when you go in that afferent arteriole, lots of blood comes in, and the efferent arteriole is really tiny. You get a back pressure in this whole capillary system, and you start to push out with that pressure. You're going to kind of bleed that blood plasma, that fluid, out of those podocytes. So it's a really cool design, right? The outlet is so small that you create the back pressure to allow the podocytes to do their job. So just an incredible design when you think about it. I really get a kick out of that. So from the Bowman's capsule, where are we going? Well, if you labeled the diagrams, honestly, you probably don't need to write this down. You go to the PCT. PCT takes you down the loop of Henle. You come up the loop of Henle to the distal convoluted tubule. At this point, we are pretty much done with reabsorption. I probably can't write reabsorption, but to reabsorb things, to bring things back in, we're pretty much done at that point. By the time you get to a collecting duct and a calyx, you are now at urine. And so we are done. It's kind of like, when is it blood plasma and when is it filtrate and when is it urine? You know, what is it? When is it what? Okay. So basically in the capillary, blood plasma. Once it's in the Bowman's capsule, filtrate, reabsorb, 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 reabsorb. Once we hit the collecting duct and reabsorption is done, we're urine. We shouldn't say it that way. It's urine. It's weird to say. We're urine. Anyways, whatever. Feel good about that? You got the diagram, so it's basically the diagram in bullet points. Not the most exciting thing ever, but reabsorption. All right, cool. So here's the diagram that's probably going to be the most similar to what you would see on your test. It's very, very similar to what you've got already. The afferent arteriole comes in, becomes a glomerulus. There's a lot of pressure there, so the blood plasma is pushed out into Bowman's capsule. The fluid then runs through the PCT, down the loop of Henle, up the loop of Henle. You'll notice there's another change in diameter of that tubing, which I think you might find interesting, probably again with more pressure. And then the distal convoluted tubule, and then the collecting duct, and it's out of there. And so that's your efferent arteriole becoming these peritubular capillaries, which eventually lead back out of your kidney. And so all the water that we reabsorb, we're just going to take back out of the kidney into, your inferior vena cava. And we'll take that up and filter through our liver and push it to our heart and, reuse that kind of stuff. So that's the nephron. And just imagine that times 2 million, you got yourself a pair of kidneys. All right, so these you already know. Afferent and efferent is arriving. Oh, sorry, Mr. McIntyre, I see your hand.
So, what does the vas erecta do? Is it just the name for those veins?
It is a different name for the peritubular capillaries when they're running straight up and down the loop of Henle. Same arteries, same job, same destination, different spot. Yeah, they just call these guys peritubular capillaries. These guys vas erecta, which is, this recta is straight, so they're the straight, vasculature. They're the same thing. These guys go around the tubes and reabsorb. These guys go around the tubes and reabsorb. Anatomists, they just like to name stuff. So, yeah. If my YouTube channel ever gets more than 6 views, somebody might type in the comments and correct me. You never know. Actually, the vas erecta has, maybe you can, here's hoping. Okay, great question. Afferent arteriole and efferent arteriole. These guys you probably already know just from our diagram. Afferent arteriole brings the blood supply into our glomerulus, and the efferent arteriole is going to drain the blood after the filtration is all done. And so the afferent arteriole is bigger, so that as blood is trying to exit out of the efferent arteriole, we do slow that blood down. There's a backup pressure in the glomerulus, and that high pressure is used to actually force our solutes out of our blood and get them into the Bowman's capsule. It takes a lot of pressure because what you basically have to beat is what's called osmotic pressure. And this is a tricky thing. Hopefully you've all taken chemistry, but essentially here's a membrane. Both sides of the membrane have water, H2O and H2O. Got it. Okay, both sides of the membrane have water. Now, what you have is a situation where if all these dots are solutes, so this is the dissolved, sodium and potassium and calcium and urea from the food you ate, water will naturally want to move to the place of higher solutes. That's called osmosis. That's just what water does. If you slice a cucumber skin off and you put salt on it, water will get sucked out of the cucumber and run down the side of the cucumber. If you're a mean jerk kid and you salt a slug, water is drawn out of that slug and he starts to foam because the water is being drawn out of him and you're killing him through dehydration. And that's osmosis. And so the tricky part of this is that basically if this is your, oh, I have one comment, hold on. It's probably about slugs. It's so fun though. It really is. Okay, so if this is your blood, we're gonna write blood. Oh wait, hold on. Haha, that's genius. Just kidding, humble brag. Okay, if this is your blood and this is your Bowman's capsule. Ooh. Okay, this is getting fun. If that's your blood and that's your Bowman's capsule, basically the water and the solutes don't want to leave. If anything, you want to suck water out of the Bowman's capsule into your blood because that's where the solutes are. So here's the trick. How do we get this out? We have to create more pressure on this side than the osmotic pressure that water naturally gives us. And so by making that efferent arteriole small and increasing the pressure on this side, we can beat osmosis and force our water and solutes through that membrane, if that kind of makes sense. And so that's what you want to do to get that water and solute over here into your Bowman's capsules. You have to create that pressure. And that's what we do, and it seems to work great. We're all still alive. Our kidneys are working fine. So that's how osmosis works. If any of you guys have a reverse osmosis machine in your house, that's what it does. It takes in hard well water with lots of dissolved salts and solutes, and across a membrane creates so much pressure that you leave the salts and solutes on one side and you force just clean water to the other side of the membrane. And then the water coming into your house is now, softer. It's not really a water softener, but it's taking ions out of it. So in my head, those two are very, very close together. But yeah, so that's how you get the, urea and uric acid and calcium and those kinds of things pushed out of your bloodstream and into Bowman's capsule. Pressure is key there. All right, so good news and the bad news. Good news is kidneys are awesome and they take a lot of our blood. The good news and the bad news here, 99% of everything we pushed out, we are going to reabsorb into our bloodstream. That's crazy. That's like, why are you even filtering? If you're going to push out blood plasma and reabsorb 99%, are we even doing anything here? Yes, we are. We're taking the dangerous stuff like ammonia and uric acid and urea, and we're getting those things out of the bloodstream. But it's actually good that we reabsorb 99% because based on the blood volume that your kidneys get, you're going to be filtering just short of 50 gallons of blood plasma a day, which is insane. Like if you think in your head, the picture of a 55-gallon drum, like from a farm and ranch setting, you know how tall a 55-gallon drum is, right? They're these big metal drums. That's like almost how much blood plasma is going through your kidneys. That's ridiculous. And when you figure we've only got like 5 liters of blood, and we must be filtering all our blood like 9 times a day or something like that. I don't know. I'm really bad at math, but seriously, like holy moly, right? That's crazy. So it's cool that we reabsorb 99%, or we would be urinating 47 gallons a day. That's not okay. That's ridiculous. Okay, that's ridiculous. So we reabsorb 99%, and we end up urinating something like a liter a day or 2 liters a day, and that's not so bad. You guys kind of have an idea of how big a liter or liter and a half is. I normally have my water bottle in class with like milliliters on it, so I can show you, but I don't have that, so I don't know. Take your hydro flask, guess at a little over half a hydro flask, maybe. I don't know. I don't own a hydro flask, but I know many students do. So I'm trying to visually gauge how much a hydro flask holds. My guess would be probably about 2 liters. Grace, what do you know?
Okay, I have a weird question.
It's a weird chapter.
Yeah, why is it that when you drink a lot of water that that it's like a lot less yellow?
Yeah, that's a great question. Does anybody want to answer it? Because I bet you guys can answer it. Nobody wants to guess? Are you sure? Because I think we talked about it yesterday. I bet you guys could guess.
I can give it a shot.
Okay, Tori, give it a shot.
So the more water you drink, the less concentrated all of like the things you're filtering out is going to be. And so if you drink less water, your urine is more concentrated. And so the more, I don't know, stuff you're filtering out is at a higher concentration.
That's perfect. That's exactly right. So,
Worse?
sorry, what's that, Peyton?
Is that why it smells worse too when you're not hydrated?
Yeah. Yeah. So the amount of protein that you're eating, right? The nitrogen from the protein that you eat is going to become urea and uric acid. Urea and uric acid is the smell that urine has. So you eat the same amount of protein every day, you make the same amount of uric acid, which is going to make the same amount of smell every day. So if you have that same amount of uric acid and urea and you've drank a cup of water, it's going to smell pretty strong. You have that same amount of urea and uric acid the next day and you drink a gallon of water, you've diluted that, like Tori said, and you're hardly going to smell it. It's going to be almost clear, right? And so that's exactly right. It is a concentration question. How much urea is in that urine? That's the question, and that's exactly right. That's not a weird question. No, that's a great question. Any other questions?
I have a question.
Yeah, Juliana, what do you know?
So if you drink a lot of water, like way more than you do usually, will you still only like excrete 1 to 2 liters per day?
That's a great question. I'd have to look it up because this is kind of the discussion we had yesterday, right? Somebody had mentioned, well, during track season, we drink so much water. And during that first few weeks, like we're just urinating all the time. And then you kind of get used to it and you stop urinating so much. And so I don't know, Juliana, in my opinion, I would, or limited experience with this, I would say probably I would guess you would urinate more. That would
Oh,
be my guess because you only need a certain amount of water in your blood to give you the blood pressure you need, if that kind of makes sense.
mhm.
So if I need to absorb from my diet, let's say, I don't know, a liter, let's say I need a liter of water a day absorbed into my bloodstream to give me the blood pressure I need. And I drink a liter and a half, I'm going to urinate out a little bit around that half liter mark, right? But if I drink a gallon, then it would make sense to me, maybe I'm wrong, that I would urinate out like 2 liters because I don't need that much extra water in my bloodstream. So that's the way I think about it. But I could be wrong. I don't know. As far as that, kind of short adaptation thing we had talked about yesterday, it's that's a pretty tough question to answer. But Mr. McIntyre's got something for us. Let's turn it over to him. What do you think, Jake?
So does sweat then directly come from the bloodstream? Like the blood,
Sweat.
like give off the water, so then your body can put the water out to cool it off? Because I was
Well,
thinking, like, if there's more water because you're sweating more, because you're working harder, then you need more water. But then if you're sweating it off, you wouldn't have to pee as much because it'd be coming out as sweat.
that's actually a really, really good thought. Yeah. Okay. Yes. So, sweat comes from your sweat glands, like we learned about in chapter 6 with the skin. So, there's a gland, right? An exocrine gland with a duct, and those cells make sweat, right? That you secrete out to cool yourself off. But those cells are nourished by capillary beds, which hold fluid, which hold blood plasma. So, to create more sweat, if you're in track practice, like the question was yesterday, you're going to be drawing more fluid through those cells and more blood from the capillaries to nourish those cells and getting rid of that extra fluid. So yeah, my hunch would be, as we're working like rough math, if I need a liter in me, but I'm also drinking a gallon, but if I sweat out an extra half liter or liter, then right, like we could bring our numbers closer together to urinating less. So that's a really good point, Jake. I like that idea. You might very well be right with that. Any other questions or comments or thoughts on that?
At what point does fluid that you drink enter your bloodstream?
Oh, sure. So remember in the digestive system, we said the stomach absorbs some water and the and the large intestine absorbed a ton of water. It kind of depends, that single drop of water, when did he get absorbed? If it's absorbed in your stomach, we're talking about, I don't know, 10 minutes, right? A short amount of time. It's in, it goes into a capillary, gets into your bloodstream, and it's in your circulatory system. If it goes into the large intestine, obviously that's a longer period of time that you would be waiting essentially, but it's going to go, that little drop of water is going to go through an endothelial cell of the large intestine via osmosis. Like there's more dissolved ions in the large intestine cells. That little water droplet goes through osmosis and gets into the cell. And then that little water droplet would go from the cell to the capillary and become part of the bloodstream. And you'd have more fluid that way. So it kind of depends on where he's absorbed. But yeah, that's a really good question. All right. Any other questions? Go once, going twice? 3 times sold? Well, I tell you what, guys, we're about at 9:30, so I think we'll probably shut it off there. I'll try to get you an email